So, building blocks. We've seen this slide a whole bunch. We've already talked about nucleotides in full. Amino acids are the building blocks of peptides and proteins. This is the conversation for today. Okay, protein structure. So just as DNA and RNA are made from polymers of nucleotides, proteins are made from covalently linked chains of amino acids. Okay, an individual amino acid is our monomer, and when they're all together in a long chain, we have our protein. Okay. The repeat units for the nucleic acids were more restricted in the sense that there were only 4. We kind of defined them by the 4 possible bases. For amino acids, we have 20. This is the basic structure of the amino acid. Okay, so for our nucleotide, we had sugar base phosphate. For our amino acid, we have amine group, we have carboxyl group, and we have some sort of side group, side chain group, in this case generically noted as R. Okay, and these are all attached to an alpha carbon. Okay, so we have some alpha carbon in the center, and attached to that will be 3 things: amine, NH3, carboxyl, COH minus, and some side chain. What is the simplest possible R group? H, perfect, just a hydrogen. Which amino acid has that? Okay, and it's okay if you didn't know that because I'm going to give them to you always. And if you did know it, aren't you sad you memorized it? Is the amino acid, so is a amino acid monomer here neutral? As a whole. Overall, we'll say yes, right? We're seeing charges listed there, but we'll say overall it's neutral, so I agree with that. Is it a dipole? Yes, this overall molecule is a dipole. We have a separation of charge. Okay, the amine group is positively charged, the carboxyl group is negatively charged. Great. All right, so now I'm going to go through some imagery of all the amino acids. Again, don't have to try to sketch them down. You don't have to memorize them. Okay, so here we're looking at the hydrophilic residues. Hydrophilic means they want to interact with water. They're able to make hydrogen bonds with water. We have some positively charged ones, okay, such as arginine, lysine. Go ahead and ignore what's going on with protonated histidine or deprotonated histidine until we get to chapter 10. And then we'll talk about it in extensive detail. Okay? It's dependent on the pH environment, whether or not the hydrogen will be on histidine. Okay? We also have negatively charged amino acids. So we have aspartate and glutamate. Okay? These are polar. They can participate in H bonds with water. They have a permanent charge on them, meaning it is most energetically favorable for them to be charged. Okay? They obviously could have the hydrogen missing. Okay, so they're no longer charged. In this case, the hydrogen could be on these groups, but that's not the lowest energy state for these molecules. Okay, so they bear permanent charge. That's their natural physiological condition. Just a reminder of the arrows. So the blue arrows here are H bond donors. They have a hydrogen to give. Red arrow here indicated H bond acceptor. Okay, and just even though this gives you your 3-letter version and your 1-letter version of all of these, again, you don't have to memorize them. Okay? You don't need to memorize the structure, 3-letter or 1-letter codes. These are residues that have polar groups. So their side chain is polar. They can still participate in bonds, hydrogen bonds with water. However, they are not permanently charged. Okay? They're neutral, but they have some ability to participate as a donor or acceptor, depending on which is which, with water. Okay. What is special about cysteine? Again, what is special about cysteine? Can form disulfide bonds, okay? In case you didn't hear that one, perfect. It's almost as if you knew it was coming. Okay, so there's a sulfur here that can participate in a disulfide bond. Disulfide bond is occurring between 2 cysteines, okay, between these 2 sulfur atoms. What type of bond is a disulfide bond? It is a true covalent bond, okay? It's a true covalent bond. It is thus stronger than some of our non-covalent interactions. Tryptophan here, I only boxed this one because tryptophan in pretty much any other list of these you'll see will be tagged as nonpolar. So I don't really know why the textbook lists it here as possibly having this little one H-bond donor. It's a really big ring group that's mostly going to participate in hydrophobic stuff. So I don't know what your book was doing. So then we also have all of our hydrophobic residues and all of our nonpolar residues. Okay? So we had glycine we mentioned was special because its side group is just that hydrogen. It's the smallest side group. What's going on with proline? So it has a ring, but all of these other rings, here's my amine group, here's my alpha carbon, here's my carboxyl, and then the side group has the ring on it. Here, both my alpha carbon, which is actually located right here, and my amine nitrogen are in the ring, okay? So that's kind of what's unique about proline, is that the nitrogen and alpha carbon are going to be in the ring, okay? Which gives protein chains more flexibility, G or P? G, okay? This is obviously much more flexible. It has such a small side group, it's not going to get in the way of other groups, less likely to have problems with steric clashes or steric repulsion. This is really, really not flexible, okay? This is the ultimate figure I think I ended up giving on the first exam last time. So this is probably the one I will give again. Okay? So you will have access to a figure that looks like this. It has them labeled as nonpolar, positive charge, negative charge, or polar. In this case, again, I've just drawn your attention to some of the unique ones. Tryptophan is again shown as nonpolar, as you will most likely see it if you just went onto the internet and searched for them.